Thank you so much, Tina. Luckily, it was all just intro stuff before that. Yeah, so these are recorded, and I have a YouTube channel that I made for this lab section, so you can watch the videos there. And I think that's in the resources list that I sent out. So in the messages to you, it should have the link to that. And then I'll try and get those up later today or tomorrow after the lab section. So yeah, Tuesday and Thursday mornings are my office hours, same link. Make a Google event and you can just put in the description the link, so it'll pop up a reminder and then you can just click and be right here. So pretty easy. Reminder that you have, in general, in a week, you have your lecture homework, and then you have your pre-lab and your post-lab. And the pre-labs are going to be due by 9 a.m. on Monday, and the post-labs are due by the end of Thursday the following week. So our first post-lab will be due next Thursday, the 15th, I think. And then you can just email me on Canvas, or message me on Canvas, or email me also to my email with any questions or concerns about this. This is a weird time, so it's a little more flexible than usual about certain concerns or if things come up. But you're lucky that this is like the second year that we're doing this and things are a little more organized this time. So are there any questions about the details for the lab section?
Yeah, so I guess received the lab kit, and I seen the video, like we're supposed to do the plantation, like
Sorry, the planting?
yeah, the planting for the like that we have to do for lab 4, I guess.
Okay.
So they said we're supposed to start either from April 2nd to today's the last day, I think. So I
Okay.
just got them, so I'm gonna try to do them today.
Awesome. Yes. Sure. Yeah, that's good to point out. Yeah, and lab kits, that's a good point also, is that, is there anybody who is abroad who does not have a lab kit? You can.
Oh, hi. I'm not abroad, but I just wanted to say, like, I also just I'm barely getting my lab kit too, so I haven't planted them yet. That's
Okay.
okay, right?
Yeah, just when you can.
Okay.
Yeah. Yeah, and so it sounds like we don't have this problem, but there are ways around, not if you don't have the lab kit. I know that they weren't able to send them abroad, so, but there's lots of ways of adapting the stuff if you don't have it. All right, so moving on. Okay, so the topic, we'll kind of dive in to the topic of the first lab, and then go over if there are any questions that you have about the pre-lab assignment, and then also we'll go over what you need to do for this lab assignment, and then you can talk about it. What would happen if the plants either didn't grow or die throughout the experiment? I think we'll find a way to work around that also. Just do your best. We know that things can be unpredictable. Yeah. Okay, so the topic of this week is discovering diversity. So some topics that we're hoping to explore during this lab is, what is biological diversity? How can we quantify diversity? How do we sample for diversity? And then kind of how do we know if we've taken enough samples? And these are really important questions if you're trying to understand diversity on a landscape scale or in any environment. And this lab kind of gives you the tools to start, inspecting these questions for yourself in whatever environment you're in. So does anybody remember from the pre-lab how we define diversity? Feel free to type it in the chat or unmute yourself. There were 2 factors.
Is it about richness and evenness of species?
Yeah, exactly. Great. Yeah, so the two things that go into diversity are the species richness and the species evenness. So we're talking about the number of species that there are. And I think in the pre-lab, they said there are two ways to classify diversity. You can just look at the number of species, but really like when we talk about a diversity index or we're talking about how diverse a site is, we're also kind of looking at the evenness. So how represented each of those species are. So their relative abundance. So you kind of practice this a little bit in the pre-lab, like looking at the diversity equation and looking at, they had the diagram with the ants and the different animals there and seeing how much richness there was and how much evenness there was around the landscape. Does anybody have any questions about these two factors? Okay, so one way to quantify diversity is this true diversity index. And I know that this is kind of a scary, I find this equation scary looking, lots of letters and numbers going on, but really the main thing that this is trying to get at here is that you have something that's quantifying the number of species. So like P1, P2, P3, think of that as like a bird, a mammal, a plant, like different species. Each P is a different species. And then the little number, the negative P1, this is the proportion. So this equation basically is encapsulating those two factors that we talked about, the richness and the evenness. And it's just an equation that can help us see how much of both are present in our site. So does this concept make sense? Did people find this challenging on the pre-lab? Do you feel like we're getting where this is going? It's a plug and chug kind of thing. You can just plug the stuff in, but I know it's kind of scary looking. And I think that there are videos. Biz2B is one of the best recorded, documented courses at Davis. So there's lots of different resources. There's videos that they've talked about this equation, and then also calculating the rarefaction plots that we'll talk about in a little bit. So you're more than welcome to come to my office hours if this is still confusing you, but also there are tons of videos to watch. So yeah, any questions about this? Okay, awesome. All right, well, before we move on to talking about the lab, were there any specific questions from the pre-lab that were confusing to folks, that they weren't quite sure how to answer them or they weren't sure about their answers? I graded all of the pre-labs and those should have been released to you. And I don't necessarily want to go into like specific people's grades. I'm not going to do that on here, but you could definitely come and talk to me and like figure out which questions you may have missed on the pre-lab and office hours.
Yeah, so I have a question. So
Yeah.
like in the pre-lab, I know you released the grade, but I couldn't see like, which like, the like which one I did wrong or which one
Yeah,
I did right.
totally. Yeah, so we don't release your graded answers, but if you come to office hours, I can
Okay.
talk to you about which questions it was, but because we're
Okay.
grading them not on Canvas, because it's the Google form,
Okay.
we don't have a good way of sending it back to everyone, but
Okay.
yeah. Let's see, I have the question. I'm just trying to see if there was one that people did really well on this lab overall. I think that something that was challenging was no, people got the diversity index calculations. I think that something that was important was that both sites had the same number of species, but then one of the sites had a higher diversity index because it had more evenness. So that was kind of the trickiest thing there, is seeing that kind of difference in how we're calculating diversity. Yeah, any other questions about the pre-labs before we move on to talking about the lab? Yeah, so we're looking at the correctness, but sorry, someone just asked how the pre-labs are graded. So yeah, we get your answers and then a key, and we're there is a little bit of flexibility. Some of them are, you can argue your way. Like I'd say that the most important thing is to be clear and concise and really get at what the question is asking. You don't have to write an essay, but you can argue what you think. Like one of the questions was on the last one, which one would you preserve? Which site, like site 1 or site 2? And either of those could have been correct. You just had to have reasoning for it. So some of them there is an exact answer, like a number. And then some of them it's like your opinion, but as long as you're using ecological concepts to base your answer in, then I'm going to give you the benefit of the doubt. So that's kind of, it's a mix of both, but yeah, just making sure you're addressing what the question is getting at, I guess. Sweet. Okay, so moving on to our first lab. Does everyone have access to the lab manual? If you don't, there's some instructions on how to get that. I had a question earlier about getting it through equitable access, and it should be included in that. You just should get a code from the bookstore if you need it. And you can download the post-lab assignment and all the lab documents from that lab manual. So on the, someone asked how to get it. If you go onto the main Canvas page, there should be instructions on how to get the Learning Tree lab manual document. Oh, you could not access it. Okay. Do you have equitable access? Yeah. Okay. So if you didn't get a code from the bookstore, you might need to contact the bookstore to get your code for accessing the lab manual materials. So you submit the post. Okay, someone's saying to check the bookshelf. Yeah, I haven't used the equitable access for material, so I'm not sure how it usually goes. So the post lab will be submitted on Canvas, and you can submit it as a Word document or as a PDF. And just a note, if you have a Mac and you use Pages, just please save it and submit your post lab as a PDF, not as a Pages document, because I have a PC, so I can't look at it. Okay, any other questions about that? And yeah, so check the bookshelf tab, reach out to the bookstore if you're still having trouble, and you can reach out to Pat Randolph, the lab coordinator, is probably your best bet. He knows everything about this lab. So you can reach out to him or to me if you're still having trouble. All right, so here it is, the exciting first lab of our quarter together. Really fun looking at diversity together. So actually apart, but together we'll learn about it. But so basically you're going to select a habitat to sample, and this is adapted so that we can do this from wherever we are in the world. Everywhere that we are, there are different habitats. So go to your yard, the weeds in the sidewalk, wherever you are, a park, your backyard. If you can't go outside, this is just, if you can go outside, please do so. If it's not safe, if you can't, if you have no access to an outdoor area, that's okay too. You can run a transect from one end of your house to the other, and you can classify objects like chairs or desks or socks as species. But really, if you can do it outside, that is much more, much more like the real life experience of doing diversity sampling. But yes, we will work with what we have here. So you were introduced to the two sampling techniques of the quadrat or the transect, and basically you're going to pick which one you want to do, and you're going to take 8 samples and you're going to count the number of species and the number of individuals of each species. So it's just like you did in the pre-lab, but in your habitat, wherever that may be. And the important thing here is if you're doing, so everyone has the lab kit, so this won't be an issue. If you don't have the lab kit, you can use string or a hose or rope or something as a transect, and you can cut out a circle, or some people have used hula hoops or what have you to do the quadrat. And you're gonna pick, so if you pick the quadrat, you're gonna see how do I kind of randomize the sampling? So you have to decide, do you turn around, close your eyes and throw the quadrat? Do you do it at regular intervals? That's up to you to figure out how to do. And then if you do the transect, you'll also figure out what interval you want to do the samples on. So you're only gonna take 8 samples of either of the ones that you pick. Then once you have your species and your number of individuals for each species, you'll calculate the diversity index for the habitat using that equation that we talked about, and then you'll create a rarefaction plot with your data. And then there's this video here for how to sample your habitat. And I can try and figure out the best place to post this if you want to have access to this video. Yeah, so we talked about things for you to think about. What are the different sampling methods? Which one do you want to use? How do you place the transect in a habitat? And then how to determine the species or an individual. So we're definitely not expecting you, if you're out there, to like know all the species by their scientific names or anything. It's just kind of like small green bug, there were 4 of them. Like grass, there were X number of individuals. And then another thing to think about, and this is kind of your discretion, and this is true as scientists, this will be your discretion if you go into research too, like how am I classifying an individual? How am I classifying if it's, if it's a piece of grass or something? Is it the whole clump of grass as an individual? Is it one blade? Is it where all the roots are connected? So these are things that you'll want to think about. Something that they say in your lab manual that I think is important here is don't spend more than 90 minutes like sampling things. You don't have to count every single blade of grass. Like, make it easy on yourself. So yeah, anything else any questions about that? As long as you're consistent, that's the most important thing. Okay, does this all make sense? Do you feel prepared to go and sample in the wide world? Yes, seeing lots of thumbs up. Yes, blossoming scientists, very awesome. Okay, then the thing will be, the other thing you'll be doing is making a rarefaction plot. So this is kind of something to stress and important is you will see so many graphs in Biz2B. There are so many graphs, and it is so important to look at the axes and what they say and what is this showing. So the number one tip I can give you for like understanding what a graph is let's start by looking at the axes and see what this graph is telling us, and that axes will tell you that. So this plot, we have number of individuals on the x-axis and the number of species on the y-axis. And these are 2 different kind of curves that you could possibly get from the rarefaction plot. And they talked, I think they talked about rarefaction plots in the pre-lab, right? Yes, okay. And basically, this is showing like, as you get more individuals, are you finding more species? And if you're getting kind of this linear reaction, like it's just going up and up and up, then you have a lot more species to find. If as you get more individuals, you see this leveling off of the number of species, then you might be close to finding most of the species in your habitat. So I believe on the post lab, there's going to be a question about, do you feel like you found all the species? And kind of look based on the curve of your rarefaction plot and then interpret based on that whether you think you've found most of the species. This is one of those questions that's like, it's all about your data and what you found, and so you just have to interpret your data and show me that you understand that, like, oh, if I have this linear reaction, I have a lot more species I could have found, because the more individuals I find, I'm still not seeing a leveling off. Whereas if you saw a leveling off, then maybe you found most of what could be found. Is there any questions about that? And we can also go over that more in office hours if after you've collected, you want to do your data collection first and then you're struggling with that. All right, so to measure the area we're doing. You don't have to measure the area. Just kind of determine yourself what the size of the area is. All right, any other questions about this? One thing that I just wanted to mention is you can ask any questions about lab topics or what we're going over in this, or pre-lab, post-lab, but for the lecture material, sorry, this is a little bit intense with the Bill Nye gif. For the lecture materials, you should go to the lecture office hours. So Dr. Gerhard Barley's office hours, or blanking on the other professor's name, but go to them to talk about the lecture material and then talk to me, or you can email Pat, or go to any of the, I believe you can go to any of the TA's office hours to ask about lab questions. So with that, that's all the material I have for today. So if there aren't any other questions, I'll hang on out here for a bit, but otherwise I'll see you next week at the same time. I have my office hours tomorrow morning, and then on Tuesday morning, turn in your pre-lab on Monday morning before 9. And then most importantly, have fun doing this. It's so cool. You get to do science wherever you are. And yeah, I'm really stoked to meet you all and excited to keep doing all these fun labs with you. So you can hang out, ask questions, or you can feel free to go and do what you got to do, work some science, magic.